Hello everyone, I hope you had a nice summer. Today I'm telling you this is my third and last talk I'm having at EDU-ARCTIC. I have to say that's been a lot of fun so far. For the ones who don't know me or haven't listened to my previous talks, my name is Kopatz. I'm a bear researcher from northern Norway. So I live close to the border to Russia, and here, me and my team, we are studying the brown bears of whole northern Europe, basically. I will have some slides from my previous talks, and some of you may have joined the talks by Julia. So, as everything in life, and specifically in biology, all is connected. So you will maybe recognize some slides from previous presentations. And I show you in this scenario what we have been doing over the last, the last 10 years of genetic monitoring of brown bears, specifically in Norway. So if you maybe have some questions in between, I think feel free to type it into the chat. Otherwise, we take them afterwards, after the presentation. A bit on the population history of brown bears in northern Europe, or specifically Norway and Sweden. You will see in a minute on a slide that actually this population is connected. So the Swedish and Norwegian population, they form one group. So, but in the late 1900s or in the 1900s, people had a different opinion about large carnivores or brown bears in specifics. They wanted to kill them, due to trophy, due to danger, or whatever was the reason. So here you have the number of bears on the y-axis. On the x you have the timeline. I picked 3 points in time to show you what actually happened in the past with the brown bears in Norway and Sweden. So in 1900, it was the political atmosphere of wiping out large carnivores because of many reasons I told you. So they were hated. There were nuisance. There was a trophy on it, so you could actually earn a lot of money by killing a bear and then sell also its parts. So there were about 3,000 bears in Norway by the time of 1900, which is a massive number considering the size of the country. Sweden had about half of it, 1,500. But then, of course, they were almost extirpated and wiped out, especially in Norway, while Sweden still had 50 years later, a number of 600 to 700 bears, while it was virtually extinct in Norway by 1950s. In the 1970s, environmental protection came on the political agenda. So conservation-minded management and politics protected large carnivores in many countries, especially in Europe. So since then, the numbers have been growing again. So in Sweden, nowadays, in 2011, it was 3,000. Nowadays, it's a bit down. The management decided to reduce the number of bears in Sweden again. So it's about 2,800 now, but we come also to that in a minute. While in Norway, the recovery or the growing of population in numbers has been really slow. That has also different reasons. So, but since 2006, the Norwegian management installed the policy of national monitoring every year of the brown bears in the country. We get to that now. Just to illustrate this figure on a map, here you have the 1860, 1870 to the 1920s, 1930s. So you have Sweden here and Norway on the western edge, coming up over Finland here, and Russia is on the other side. The white area presents a region where the bears actually are permanently present, or were permanently present, while this gray area where bears were not, or occasionally present, or were actually not existent. So you can see over time, during the times of extirpation and killing and hunting, that this area shrank down, or this area was growing. Mainly noting in Norway being almost wiped out of bears by the 1930s. You have a small area here in Oslo, and yeah, that's it. And Sweden was only half of it was maybe with bear country. But also, these numbers you have to take with a grain of salt. This is historical data, so it cannot be really proven. So it's also a lot of anecdotal wisdom in it. So, but at least gives you an idea. Okay, talking about conservation-minded politics and management, the Berne Convention. So 1986, the Berne Convention got into play, which actually binds legally all participating member countries to protect the flora and fauna, especially the protected species, and the habitat around them. So Norway has been signing it, as a lot of other countries, and it legally binds us to monitor our fauna, flora, and actually also actively protect it. I know there's a lot of, it's never that really clear, but this is the baseline. So to set the scene, this is the distribution of brown bears in Europe as it is today. We have Europe here, we have Scandinavia and Fennoscandia with Russia here. The red areas represent areas where brown bears are permanently present, with, let's say, orange or skin color tones where they occasionally are present. So you have this really big dots here in Sweden where you have a lot of bears. And already here you can see, if you pay attention to Norway, that you find this red areas in Norway only on a few places. So I already can hint to you that the Norwegian brown bear population is by most far a trans-border population. So the bears coming to Norway, a lot of them originate from Sweden. While on the other hand, up here, where I live, this dots, that's where I live and work, this population is mainly connected to the Finnish and especially the Russian one on the east. You notice the Russia is blank. That doesn't mean that there's no bears, but Russia has approximately the largest brown bear population in the world. But for us, it's a big black box. So there's no really precise thing available. The estimates are very rough. We know only that the population is massively large. So just to bring you up to speed, in Norway there's about 150 to 200 brown bears approximately. So this is a minimum number. In Sweden there's nowadays about 2,800. Finland has about 2,000. As I said, Russia probably much far over 100,000 brown bears. Probably European Russia something about 50 to 60,000. And then Estonia with this very small country with approximately 700 brown bears. So very, very surprising. And then of course you may be aware of the situation in Central or Southern Europe where we have a lot of endangered populations in the Pyrenees and the Cantabria Mountains. This is a very massive population, the Dinaric Balkan population, and of course Romania and Slovakia. Yes, but how do you monitor? So as I said, the Berne Convention obliged us to monitor brown bears and take care of them. So very important information on the monitoring is basically how many bears are there? How do we get to this information? We have the numbers, we have the distribution. So how many bears are there? Where are there? What are the areas that they're using? How much room does a bear actually need? Is the number, if you look at across time, if you monitor regularly, that's the point of it. Is the number increasing or decreasing? So if it's decreasing, what does it actually mean? Is it increasing? What does it mean for the management? Will there be more conflicts with humans, for instance? Especially for survival, important is the reproduction. So how many numbers of litters, meaning young cubs, how many young bears are born every year? How many die? And especially also reproduction is the male-female proportion. So what is the sex ratio here? Do we have more females than males or the other way around? All this is very crucial information if you want to manage a population and want to decide how to deal with it. But what is problematic with the brown bear? It's an elusive species. That means it's not easy to be seen. Elusive means it avoids humans. In Europe, the brown bear tends to avoid humans. So normally when you see a brown bear, you consider yourself very lucky, especially in Northern Europe. You might see them running away most of the time. But yeah, it's very difficult to detect, especially by eye. So that's where the so-called non-invasive genetic sampling comes into play. So in the 1990s, these methods were developed, and until today, they have been massively improved. So they became very, very reliable. So what we here see is a dropping or shit of a bear, which contains a lot of information for us researchers, managers, everybody who's basically interested in that. So it's actually a very simple method. You just have to collect the shit, send it in, and analyze it. So you get a lot of information out about it. So traditional monitoring methods like track count or observation, as you see here, like when you see a bear, are also still very important, but they are not as reliable or as specific as the DNA method. So of course you can see tracks, you can probably get an idea about how many bears are in an area, but you don't know which bears specifically. Also you could record, which is still be done, like damage or predation reports. So if livestock, a cow was or sheep was killed by a bear, of course all this information will be tracked and recorded. But since 2006, Norway is using non-invasive genetic sampling, meaning the collection of droppings, shit, hair, or other biological samples. And the keyword here is biological samples. This allows us certain identification of the individual which actually leaves the mark here. And this leads us to information about if it's a male or female. If we collect all the data together, I will show you in a minute all these maps, like the movement patterns, and we can even go further and reconstruct pedigrees or family trees, which is where's one family living and not. And what is very important, and I cannot stress this point enough, is this local engagement and citizen science point of this method. So it's not us who is collecting these samples, it's everyone. It's mainly, of course, hunters and people hiking out in the nature or berry pickers. But this immediately connects them to the whole system of monitoring. So people get literally locally engaged. So they actually have an interest and showing interest and wanting to know more about the bears in the area, which also helps to protect them and, yeah, simply to monitor them. So this is a really, really cool method. And of course, along the way, it's not only that they can send in the samples. Later on, they can find this information in the internet. We have a website called Kopatz, where you can go in and check later on exactly the sample you sent in. Has it been identified, or was it maybe a negative sample due to other reasons? I also come to in a minute. So it's very crucial to keep the people who actually invest their time or make an effort be informed and be rewarded with the information afterwards. This is a very, very critical point. So this is from Julia. Maybe you have joined Julia's talk in the past about DNA and bear poop. So for these people, it's probably very easy, but who haven't seen it, they might struggle a bit with here. But maybe can you distinguish which one of these A, B, C, or D is bear poop? Maybe you can answer quickly in the chat. Yeah, everyone is correct here, I see. It's indeed C. So this is a dropping mainly containing of blueberry from the autumn. So the bears, of course, eat a lot of berries to gain fat for the winter. The first one was moose. The B one was fox. It has this typical canine spine here, which you can also see in many dogs. And the D was a mountain hare. Let me note the bear is an omnivore. So the shit of a bear doesn't look all through a year, not like this. This is a typical autumn shit you find. It may look totally different in summer and spring, when maybe it's ants, for instance, fresh leaves, or meat from carcasses they may gain. But that also leads to different results in DNA results, which I also will present you very soon. Yeah, thank you for answering that. Very quickly, a reminder of this has also been part of Julia's talk, how it works to show you. So we get the samples here, It can be from the wildlife crime scene, like here, and some blood stain. It can be from a shot bear. Some bears are legally shot, especially when they're causing problems, destroying property or killing sheep, or just hunters and berry pickers or hikers sent in feces. But we also collect hair samples from fences or systematically hair trapping. So these are sent to the lab. They are registered, analyzed by our staff. With extracted DNA, genotype it, and then we have an identification or a hit on a certain individual. And then we compile the results in the end, plot them on the map, put them together, trying to interpret them, like putting the whole picture together, which hopefully makes sense in the end. And then we report that to the public and our agencies. So let's come to some results. This is from last year's collecting in 2016, so they were analyzed throughout the last winter. There were like 978 samples collected, so roughly over the years it's always something between 1,000 1,500 samples we get to be analyzed. The majority of them is feces, shit samples. Then we have some amount of hair samples, 240 in this case, and 8 tissue samples, probably from some shot bears. The brown bear management in Norway works like this. There's different so-called predator zones. So I'm, for instance, in predator zone 8 is Troms and Finnmark up here. And then you have other ones a bit more diverse in the south. But already by the number of collected samples, so now we get to the results. You have this districts or zones. You have the number of samples, and the colors indicate a bit what kind of sample is what. So the orange is hair, and the blue is the feces or shit. And you already see 3 districts popping out with quite a large amount of samples, while the other ones were quite low, except this one maybe it's a bit higher from Troms. But we have Finnmark up here north with 270 samples. Oh yeah, welcome to Sophia. They're joining us late. And Nord-Trøndelag and Hedmark. So Hedmark is here in the south. Nord-Trøndelag is about here, and Finnmark up here, yes. You will see it also on the map, and you also saw it in the previous map before. You kind of got an idea where the distribution of bears was. So, but this is like the baseline of information we start with. So the number of samples is already a good indication. So it's probably a bit more bears here than in the other areas. And indeed, that's the case. If you look at the unique genotypes, how we call it, so when we identify the bears, how many bears are actually in this. For instance, here we have 296 samples, so how many individual bears we have here? It's 46 in Hedmark, down here. We found 29 individual bears in Trøndelag, which is here, and at Finnmark, here it's 35. So you can see like Hedmark and Finnmark combined already 50% of the bears identified in Norway. So this is a strong hint that these counties or districts house most of the bears. So there are some bears going around. And you have Nord-Trøndelag, which is a bit in the middle with 23%, and then you have Troms here with 8 and 2%, and Norland, which is here, and Sør-Trøndelag, which is somewhere here. So now we know how many bears are. This is the minimum number, because we don't detect all the bears. This is impossible because you don't find the shit or the sample of every bear. There's a lot of influences on that. I'll also come to that in a minute. But it gives you already a very good overview where the bears are and probably how many are there. There are statistical methods, and of course to extrapolate, to estimate the populations, but we don't go into that today. This is statistics. So if you plot the results on a map divided by female bears and male bears, you get already quite a good idea, confirmed a bit by the results we saw before with the bar plots, where the bears are and how they are distributed. So you notice actually also a difference between the males and the females. First, we got a bit more males than females. And you see also that they're a bit differently distributed. So the males seem to be spread out a bit more, and while the females seem to be a bit more concentrated in some areas. Again, you see it's everything along the borders to different countries. But this is, you can explain by the biology of these brown bears, while males are actually the ones who are dispersing. So they leave their home area after they are grown up or have a certain age, while the females tend to stay in the area where they were born. This has simple causes like avoiding of inbreeding, for instance. So that's why the males move away to search for new areas and especially a new female to breed with. So this is also a very interesting information. You see the males dispersing and the males being very concentrated. So this has very strong indications for the management, especially the females, which are responsible or very crucial in reproduction. So these areas should be of special consideration for the game management when it comes, for instance, to shooting or hunting. So every year a report like this containing this information and other is produced and sent to the agencies, and that's where the decisions will be made and the population monitored. So we have for every year now this kind of information, and then when it gets really interesting is like the trends. So how does the population actually develop across time? So here I picked a specific time frame from 2009 to 2016. Again, I remind you this is the minimum number, so the total estimate probably looks a bit different, but in total the trend will be probably the same. We have the number of bears here. Well, in 2009 and 2010, we have 166 bears detected. In 2015, 2016, 128, 125. So this is very interesting. So there's a clearly negative trend of individuals identified while monitoring. So I can already spoil you, this is nothing to be really concerned of because there's many reasons which can affect that. And it's probably not most likely not a biological reason here. So there's nothing to worry about. But I explain you now what it is. So what we have to do is look at the data more specifically. For instance, if you look at the hunting districts or the predator districts, how has it developed here? So like you have head mark here from 45 to 43, quite stable. But then you have notoriously 45 to 19. This is interesting. The number has been going down. Notoriously from 9 to 0, 5 to 1 in Uppland, which is also in the south, where you see the numbers. Also, you see first, you get a feeling of what kind of numbers we are dealing with. Of course, if you there's a few more samples collected, or the numbers are a bit different, the differences look already much larger. But still, this is quite of a difference from 45 getting down to 19. The same for Finnmark. We have 32 in the beginning, then went up to 50. Then we have a sudden drop of 34 individuals in 2015, and then it went up again. So you see, also this all have kind of negative trends in the estimates, in the detection of bears. So before I come into the reasons, maybe you have an idea already what could be the reason. So the very quick quiz. So what do you think might influence this result or might have affected this monitoring result? Is it A, maybe there are simply less bears in the area, so the number of bears? Maybe the number of samples is also crucial here, so maybe there are less samples coming in. Then connected to that is maybe people don't care that much anymore, or there were not that many people in the field because the weather was bad and therefore less samples were collected. Maybe it's also the size of the area. So maybe some areas were neglected this year, or that in that specific years where there were low number of individuals or samples collected. Also, there might be an influence connected to the size of the area, is the access of the area. So if you have roads and paths, of course, more people are in the area which can collect, who can collect the samples. And maybe also the storage of the samples can be affected here, because we talk about shape samples. I didn't go into it, but maybe if you looked at Julia's talk, and I think I mentioned in my previous talks, we're talking about biological sample here. It's DNA, it's a molecule which can actually be destroyed if handled incorrectly. So what do you think? Do you have any idea of if this something is of more importance, or are there several factors? Maybe you can give me a quick reply in the chat. Yeah, you may also discuss this a bit later. I see the time is running, so in order to have time a bit for questions, I just move on. I can just tell you it's everything of it and more. So I try to pick out the main ones which come immediately to mind when it comes to sampling and the work in the field, but also there's other factors later on, like the DNA analysis, which can be also be of importance if you don't have a good system set up. So it's really all of it. So it's reason. If you have low number of collected samples, I will show you examples also in a minute. If you have not enough samples collected, you will not get a proper picture of how many bears are in an area. If you have uneven spatial coverage, that means like you have only one area sampled and the other one not, or not that quick, not that sufficiently, you also get a biased picture. If there's less people collecting samples, this affects all of it. I mean, you get lower samples sent in. You get much worse information out of it. But also, we notice in many monitoring projects that the activity of bears is highly important. They might move to some other areas depending on weather and food availability. So if it's like a very bad year for berries, we also get less samples collected because the bears are maybe in some other areas where they find food. Then, depending also on weather, and that means also the storage point I made, is the low number of functioning samples. So not all samples we get in, we get DNA out of it, or we are able to identify properly an individual. As I said, DNA is a molecule. It's actually a very stable molecule, but also it cannot stand, for instance, fluctuating temperatures and sunlight. It's very, very crucial for the survival of a DNA molecule. And of course, maybe it's just the case there's fewer bears, because we know now from Sweden there are fewer bears nowadays because they were shot. The management decided to keep the numbers lower, so nowadays there are only 2,800 bears, formerly around 3,000. To show you some examples, we come to the commune I live in Finnmark, up here in the north. You can see just quickly, like this is Svanoft. I'm sitting in this building, which is here. We have this nicely water river. You see it's quite a lot of trees for that far north, but we have some kind of microclimate. So this is located in this stretch of land between Finland and Russia. This is Russia on the other side. So if we have Finnmark, and then we have, for instance, some few municipalities. I live in this municipality called Sør-Varanger, which is basically here. And you can already see this is the number of brown bears detected throughout the different years. We don't have to go much into details, but you see the Sør-Varanger is popping out. So we have a lot of bears here. But also here, we have, for instance, this drop in 2014, I believe it was, yeah. Because we have a general increasing trend of bears, normally also a very stabilized population, but every now and then there's a bit of a drop. So there's much less bears in these other municipalities like Kertekane or Karasjok, but we concentrate on this one here. So it's a lot of information, I'll bring it through it. So here again it's the same information. This is the number of bears along the year and how many there are, but we have the drop of 23 in 2014, 2015 again 43 bears. Here we plotted the samples or locations of the results on the map. So here again you have Sør-Varanger. Here's Russia, here's Finland. In 2014, there were only 192 samples, while in 2015, we had 699 samples. So massively. But that's also due to the fact that we had a hair trapping project going on in this area, so we had a lot of more sampling effort going on in the area. But you also see these areas with the circle were sampled in 2015. So we catched a lot of more bears during that time. So what you basically see is here that the effort, like having a hair trapping project, having more sampling in some areas, or going people into areas and collect samples, has a clear effect on the outcome, as has the area, if you neglect an area. So that all means that actually if you want to have a comprehensive information of bears in an area, you have to have an even sampling effort. It doesn't mean that every year you have to have 700 samples, but maybe 300 are enough, but evenly distributed and collected from every area. It is very challenging, probably virtually also impossible to have it every year, but that is the consequence or the conclusion you can draw out of it. So why is the sample non-functional? I just mentioned it as well. I mean, I said to you, like warm temperatures and sunlight are really the killer of the DNA here. So if we have very hot, dry summers or with fluctuating temperatures and rain in between, this is not very good for the DNA to survive out in the nature. So but there's also other factors, like when you have shit or feces, it really influences the outcome, what the bear has been eating. So a lot of fat, like salmon, or if somebody is feeding on moose carcass, it's a lot. We have problems to get the DNA out of it. Or especially the DNA amplified, as we call it, because we have to make copies of the DNA in terms to analyze it. The same with grass. We don't know really yet, but there's some kind of enzyme, enzyme in the grass, which kind of is an inhibitor, as we call it, like it kind of makes it difficult to copy the DNA out of the feces. So that's why, for instance, you saw the blueberry. That works excellent from that time, because the blueberry, there's no inhibitors or any problems getting DNA out of it, but also it comes from the autumn. So the autumn is characterized, it's already very cool, it's dark some of the time, like in summer, we have midnight sun, coming back to sunlight, we have 24 hours of sunlight, so this all can kill the DNA up here. And of course, if we have the wrong species, you get a negative results for most of the things. So if it's not a bear, the sample can be also non-functional. The same applies to the hair. So if you have the wrong species, it will not work. Warm temperatures and sunlight also killing the hair, the DNA analysis. Problem could be if the hair has no root. I don't know if you take out a hair of yours, you may see a root, and that's where the cell is located and where we get the DNA out of it. So, for instance, in the beginning, some people, when they got hair from a bear, they simply use scissors. That will not work. So you have to really pull it out. Or you have too few hairs. Sometimes it's in combination with the temperature and sunlight, if the DNA is already bad and you have too few hairs, it will not work. So that's what we call a success rate. So as I said, you will not get 100% of all the samples analyzed. There's always a bit of problem like storage and so on. So, but we have success rates of between 60 and 80%, which is quite high. I mean, in the past, when this method was developed, the success rates were much, much lower. So, for instance, you can see all the samples collected in Sør-Varanger and in the other parts of Finnmark, and here are the positive samples. You see, there's a bit of a difference here. That's why you also need sufficient samples from different areas to get a good picture of bears. So what does it all mean? I mean, we do the monitoring for us. We want to know how many bears are in a country, how can we deal it, how can we protect it. Because when humans and wildlife meet, there's always a lot of potential for conflicts. We know that. So humans are expanding ever more. I mean, with climate change, the Arctic is coming exposed to so many people nowadays. People are living where they never lived before. So, but on the other hand, also the numbers, especially in Northern Europe, have been going up. So it's a bit, it's a recipe for more and more conflicts. So that's why this information is very, very crucial to keep kind of control of what is actually going on. Just to very hint how the management is actually. I mean, if you do wildlife management, you have to have a goal. So a goal is either to wipe out the bear, like it has been done in the past, or you want to maintain it, keep it stable, or maybe even for some endangered species like the polar fox, for instance, you want the population to grow. So that's why you have to adjust hunting licenses and protection measures. So, but for the brown bears, the goal is to have 13 litter per year. So it's allowed to have 13 young bears in the whole of Norway. I leave you with this information. You can interpret as you may. But so bears are allowed to have 6 litters in this vast area of Finnmark and Troms, 1 litter in Norway, in Norland, 3 litters in mid-Norway, which is the Trøndelag area, and Hedmark is allowed to have 3 litters. That's the management goal. I can tell you I didn't put it in, but the reproduction is below that. It's about 5 to 6 litters per year are detected in Norway nowadays. So all of this we use also, as I said, with this uneven sampling, just to finalize this talk. We use, of course, when you have very specific tasks for the management. I think I have shown this picture before. This is a hair trapping project we have done here in this valley. There's a farm here. There was a bears observed in the area, and people were concerned that these bears would kill livestock. So we set up hair traps, like a systematical sampling of the area. So we divide it into grids. We have 2 areas, like a control area further down the valley in the forest. We put hair traps up there, so it's not much time to explain it anymore anyway, but we collect hair in these squares, so we have a systematical division of the area, and did it for 4 weeks. And then we got a very good idea of how many bears are actually in the area. And it showed in the end, the conclusion was, the bears around the farm, they never stayed there. There was only a bear detected in 2 consecutive weeks, but they moved away after that. While in this control area, we also have all the females, you see it in red dots, they stayed in this area. So they felt at home there, but it seems that bears around the farm didn't stay there for long, and it was never the same bear. So that's how we actually utilize this information we got before, area, samples, and so on and so forth. I think that pretty much wraps it up. I want to thank you, and I want to thank the EDU-ARCTIC team to be being part of it. It was really a pleasure. But now I'm available for your questions.